So in this last video, I wanted to come back again to the process of designing and cloning guide RNAs. And in the course, we used a system that was recently in 2022 published in Nature Communications. You see the title here, and it's actually a very nice system that allows multiplex base editing in Pseudomonas and other bacteria that are compatible with the vector system. Yes, so you will find in my slides a slide, this slide, and this states this is just a citation, a copy paste from the paper. So a detailed protocol to construct the multiplex guide RNAs is presented in supplementary methods, and this is actually true. So there is a very nice protocol there, but for the sake of kind of dissecting sequences, understanding sequences, I wanted to briefly, to show you a manual guide RNA design here. And what you need to use the system are actually two different plasmids, at least two. So the PEX-128 is just a PCR template from which you will amplify the guide RNAs. And the PMBX6, this is one of the different editor plasmids from this paper, contains the whole editing system and the gentamicin resistance gene in this case. So we can get these sequences from Edgene. And as I have shown you before, we always have these sequences that are provided by Edgene. So it's now a full sequence, which I downloaded. And there is also here a sequence provided by the authors. I downloaded both of them, and they're a bit different. So there are also differences at the nucleotide level, but most importantly, the annotations are different. Yeah. So if I just zoom, you see that looks quite different compared to this. And for the sake of simplicity, I will now stay with the Edgene version. So this one has most importantly a number of oligonucleotides, which of course is not bad at all, but still. So let's quickly go through the features of this. So we have a gentamicin resistance here. This is for positive selection of transformed bacteria. We have a first origin of replication for E. coli, so a high copy number origin of replication, and a second origin of replication for replication in Pseudomonas bacteria. So you see here, it's only this little piece that is annotated, and here as well, no difference. And then we have here a multi-cloning site. So there are two BSA1 sites which are flanking a GFP gene. The GFP gene is not annotated in this version. You can see it here, MSFGFP. But I also confirmed that it is there. So the empty plasmid, when transformed into E. coli, will give GFP positive clones, while once we replace this cassette for our insert, so actually a guide RNA, then the clones will be non-GFP, GFP negative. Then there is CSY4, so there's an RNA endonuclease, and this RNA endonuclease cleaves RNA following a specific stem loop sequence. This is under control of the TRC promoter, so that's a hybrid between tryptophan and LUG promoters and a quite strong and constitutive promoter. Furthermore, we have then our base editor, so ApoBec is a cytidine deaminase, the D10A version of Cas9, which only cleaves one strand, and the uridyl glycosylase inhibitor, which uracil DNA glycosylase inhibitor, which will inhibit the base excision repair pathway in the bacteria, thereby increasing editing efficiencies. Yeah, so basically what we have to do is to put our guide RNA here. We see that there is a gRNA scaffold annotated here by Edgene, but we don't care about this, so I might as well just delete this, because we will clone two full guide RNAs with respect to scaffold and CSY4 recognition sites into this place. So what we need to know first are the cloning overhangs. So there's a GTGG and a GTTT. Okay, so you can maybe note that somewhere for later on. Then let's just look at the PEX-128. So we have many different features here. Something that is called backbone. And a whole lot of oligonucleotides. So Once again, maybe I take also the edge version for this one in terms of annotations. Um, so here we have a guideRNA scaffold now indicated, as well as a CSY4 site. And we don't have all these primers. So let's just go with this one. So guideRNA scaffold, so I told you, well, it's specific for different nucleases. So Cas12 will have a different scaffold than Cas9, but the one for Streptococcus pyogenes, Cas9, it starts with this GTTT, and then it continues. It's in total approximately 70 nucleotides, a bit more. And then here in this system, it is followed by a CSY4 site. So if we just concatenate several ones, they will be cut by a CSY4 in between, and then we will have individual guide RNA. So this is our multiplexing system, or the multiplexing system in this paper, to say it better. So the 20 nucleotides here, this will program the Cas9 for its target, while these 70-something nucleotides are required for the integration of the RNA molecule into the Cas9 protein. So basically what we need to do is to put the sequence we want here, and to make several PCR amplicons that we concatenate into the PMBEC6 plasmid. So what can we take as a target? You can use this magnetotactic bacteria maybe. So normally I like to download bacterial genomes from Ensemble. This is not really possible because it's not present on this website for the magnetosporidium, but we can just forgot that we had this, the P. aeruginosa genome that I downloaded earlier today. We just use 2 random genes, maybe DNA A and DNA N, and disrupt these for fun by introducing premature stop codons. So if you think back into what we discussed about base editing and about Cas9, so we need a codon that can be edited, and it needs to be in the right distance to a PAM sequence to finally fall within the editing window. Yeah, so of course doing this manually is a bit annoying. But what we can do is look for Q codons. These are the easiest to find. CAG, here we have one, and then see if we have a PAM sequence for Cas9 on the right-hand side, which here, so we have 2 Q codons. I mean, we have a PAM here, but this is way too close, and then there is none. So these would not be, at least with a classical Cas9, this would not be possible. We have 2 more Qs here. And now we have a PAM sequence here. So if I select the 20 nucleotides upstream of the PAM, this will put this C here in position 1, 2, 3, 4, 5, 6, 6. Yeah, so this would be actually perfect. It would be right in the middle of the editing window. So this should be very efficient normally. So we could use this. So I mark this here as a PAM. And I mark the 20 upstream as guide DNA A1. and call this, there we go. So this would introduce a C2T exchange here. We get a TAA. This is a stop codon. The gene is normally dead, should not be working anymore. If we do this, we maybe should look at that we don't have an additional initiation codon just downstream, because now I'm just severing the first maybe 20 amino acids. But normally it should be fine. If we look into DNAN, here there is also a CAA. Then there is no PAM sequence, so it's not possible here. Too close. This one here. So this would put the C that we target into position number 2, and it is preceded by a G, so this is not optimal. So let's see what else we can find. This should do the job. We have one in position 4, another one in position 7, so we have even 2 possible targets. So that should be fine. So this would be our guide for DNAN. So how does this work now? So if we want to use the PEX 128, where is it? Here. To now generate respective guide RNAs by PCR, we basically need to introduce these 20 base pairs that I have selected here in this part. We need a cloning overhang, the CSY4 site, and so on. So we just have to make PCR primers. So for the sake, to change this back to a C, otherwise it's not going to work. So for now, I will do it like this. I insert this sequence here. And just briefly save this as guide DNA A1. Okay, just close this because now we have this wrong recurrency. So I'm working in this file, also have just saved this one. So I save this one, which I did not want to modify. And I open up this one again. And just turning on the annotations again. Okay, so here I now have my guide RNA sequence pasted. And so from everything that I said about cloning before, here we already have the right overhang. We have a BSA1 site. So maybe we want to add like 2 T's in front. So this would be my first PCR primer, basically. I just have to add now something that can make it hybridize on the template. So let's take these 20 nucleotides. So my PCR primer would be all this. And my PCR will run into this. So we have the CSY4 site, and now I need to make a reverse PCR primer. So let's say I want to I can make it run until here. And these last 4 bases, so I leave them now, as we can make a new overhang. All this basically is just vector backbone. It will not be part of our PCR product, so I delete it. So I said in the beginning we have GTGG and GTTTT as overhangs. So here I would now have CTAA, so it would be different and would be okay to just leave this as a new overhang. Why not? So I could make a PCR primer until there, so it has to contain the overhang and then it has to contain a BSA1 site. So this here. And the primer has to, of course, go until the very end. So I just say 200. This will put it until the very end. So this would be now my primer number 10. And with these 2 primers, I could amplify my first guide RNA from the template that we had. So the template contains all this. And with the primers, we add basically the 20 nucleotides that program this guide RNA. And we also add the restriction adapters. So this is guide RNA number 1. And now I have to make guide RNA number 2. So this was in DNAN, the second one. And what we have now, so I don't need to do much. Oops, wrong thing to do. I basically have to change this for these 20 nucleotides from the other gene. I have to change the overhang. So it now has to fit to the 3' overhang of the first PCR product. So this has to become a CTAA. Taking small letters. And now this will not be primer number 9, it will be number 11. And on the 3' end, so the primer can pretty much stay the same. I just need to change the overhang. So we said in the very beginning, GAGG, GTTT. So this has to be a GTTT, GTTT. And now it should be fine. And it's primer number 12. And now this would be, I save a new copy. Yeah. So this is not DNA A, this is DNA N. And guide RNA 2. Again, if I now confirm that I want to save, I will have 2 identical files that correspond to the design of DNA N. So this is a bit weird. You should not make this mistake to press save, or you will be surprised by the result. So now I have 2 guide RNAs. This would be my 2 PCR products in the lab, and I can actually place them into the PMBAC6 by BSA1 cloning. Always define plasmid first, open it up. Well, can make it linear, then it does make more sense. Also, it doesn't change. So now it looks as usually. And I can clone everything together, and you see that my pieces, they come in the right order and in the right orientation. So now if I clone this, what I get here, if we look in detail, is actually this TRC promoter, which will also drive, so it drives here expression of the CSY4, but it also drives expression of the guide RNAs. So with guide RNA number 1, starting with GTGG, the 20 nucleotides that we have added. So if I just play with the annotation fields, we should see the scaffold now appearing. And then the scaffold of the guide RNA that starts with GTTT. And then once again, we have the protein binding site, so the CSY4 site here. So this is where I think here, sorry, 1 second. Oui, bonjour. Yes, so sorry, I was interrupted by a phone call. So I was just in the process of showing you the final construct we have. So we have cloned these two guide RNAs. Oops, trying to move the zoom elements. So we have cloned these two guide RNAs. We have the promoter driving the guide RNA expression, CSY4 site. And then I have just used the time to look this up. So here's a scheme of CSY4 cleavage. So we have a GGCAG. So CSY4 will cleave just after this. And then we have the first cloning overhang, the second 20 nucleotides now programming the second guide RNA on the DNA end target. Again, the scaffold starting with GTTT, a second CSY4 site. Then in the plasmid, so this is rather a construction artifact, I guess, we have another gRNA scaffold, which now is not important for anything. So basically, our two mature guide RNAs will correspond to something like this and something like this. And these will now direct Cas9, our base editor based on Cas9, to the respective positions to achieve base editing. So just as one last little demonstration, so what will happen? I don't, of course, do not have the sequencing files of DNA N or A, but this. So here we have a sequencing file for some mutants we generated in a gene called PHLF, I think. Yeah, PHLF in Pseudomonas. And we have used two guide RNAs, I think. Well, maybe only one. We've used only one guide RNA. You see that we quite often get a number of exchanges. And now you could wonder why this is a G to A exchange. So this was a guide RNA that was on the other strand and that now leads to C to T exchanges on the strand that we don't see, and therefore to G to A exchanges on the strand we see. So normally there should have been a tryptophan that was edited here, a tryptophan codon. So here we have the two tryptophan codons, TGG, TGG. And if we change the G's to A's, then we come to TAA. And you see that actually, so this was, I think, the same guide RNA, the same construct that was transformed into 4 different strains of closely related Pseudomonas. And you see that we have basically in all 4 tested clones now respective stop codons. So in some cases we have 2 stop codons, and in some cases only 1. But in both cases, the gene will be disrupted, and we will have a mutant in the first place. Yeah, so this is sequencing of a PCR product with primers that are somewhere maybe here, and then sequencing with one of these two. So with base editing, you can actually really fast make such mutations because the cloning of the guide RNA is no work at all. And then the selection of mutants is also very quick and straightforward because normally the efficiency is above 90%. So you basically don't only need to sequence 1 or 2 clones. Okay. So with that, I think I said everything that I wanted to say. Just look at my PowerPoint once again. Yeah. So maybe just very quickly. So as one last thing, just share again. Oh, this will only take 1 minute. So we of course do not normally need to look for editable codons. We have of course websites that can do this for us. We have here one that should normally now do the job. So I just use the DNA DNA. So I just copy pasted the sequence from CLC into this this one web tool. So Cas9 variant. We have a Cas9 NGG in the system that I presented. We have an ApoBac. So yeah, base editing with ApoBac. We have a NGG PAM. We have an editing window that goes from position 2 to position 9, I think. This should be maybe double checked. So I'm not sure now. So yeah, let's work with 2 to 9. Guide RNA length of 20, which is fine. And we don't have a genome for off-target analysis right now because I'm not motivated to provide one. The coding sequence does not start with an ATG. Please check. So what do? It's a bacterial gene, so it simply doesn't start with ATG. But since the web mask, this mask does want an ATG, we just add one, and now it should do the analysis within a few seconds. Hopefully. Let's double our chances to have at least one working search. So here, this is in the other way around, so it must be 11 to 19, I think. We have, again, a base editor. We need our sequence here. Oh. So the only thing that changes is the way they assign the positions. Yeah. So of course, the editing window itself doesn't change with the web service. My sequence is too long. So I will make my sequence shorter. Still too long. So while this is still analyzing, normally I think it's just not working, it's offline. This one is very fast. So if we want to induce stop codons, we should look for this here, termination in this column where the amino acid sequence after editing is displayed. So can just look in a diverse inverse manner whether I find the one that I used. So it's this one here. So we can very quickly also select from these guide RNAs. You don't have to do this manually to count nucleotides to find your editing window and so on and so on. Okay, so that would be it for me. I hope it helps if you want to use these things. Thanks a lot for attention. Bye.